everyone. My name is Megan Johnston, and this summer I had the opportunity to conduct research in Dr. Klaus Van Wyck's lab under the guidance of my mentor, Marissa. My project focused on two proteins, UVR2 and UVR3, trying to understand the potential role that they play in the CLIP protease system, a system which is key to protein degradation and maintaining proteostasis in chloroplasts. So what is proteostasis? Proteostasis is the balance of protein synthesis and protein degradation, so that the right protein is at the right place at the right time to carry out functions that are essential to plant health and growth. My lab focuses specifically on the protein degradation aspect of proteostasis. A lot of what we know about protein degradation in chloroplasts comes from what we know about it in bacteria. This is because mitochondria and chloroplasts come from endosymbiosis events, meaning that bacteria used to have a symbiotic relationship with cyanobacteria and aerobic bacteria until they were eventually enveloped into eukaryotic cells, creating what we now know today as chloroplasts and mitochondria. In my lab, we study the chloroplasts when it comes to protein degradation. The most abundant protein degradation machinery in the chloroplast is the CLIP protease system. This system is also conserved in bacteria, mitochondria, and other higher plant plastids due to endosymbiosis, as I just mentioned. This system consists of several components. So you have the CLIP protease core, which degrades the substrate. You have the chaperones, such as CLIP-C1 and CLIP-C2, which unfolds the substrate and feeds it into the protease core. And you also have adapters such as CLIP-F and CLIP-S, which recruit the substrates to the chaperone and aids in substrate selection. So how does the CLIP protease system work? To show you this, I'd like for you to first imagine that you're inside of a chloroplast. So here we have a protein which is in the stroma of the chloroplast. This protein undergoes some kind of post-translational modification, making it a degron, which the adapters recognize as a substrate. Proteins may be targeted for degradation if they're damaged, misfolded, aggregated, or just not of use anymore. The degron is a signal for the adapters to take the substrate to the chaperone to begin the degradation process. Different adapters recognize different degradation signals in bacteria, aiding in the selection of substrates, as well as the variety of substrates which this system can degrade. So once the substrate and the adapter bind to the chaperone associates with the protease core. Then the chaperone begins to unfold the substrate, feeding it into the protease core. The protease core then carries out protein degradation, releasing 6 to 10 amino acid peptide products. Once the degradation process is complete, the chaperone and the protease core disassemble and go their separate ways. So as adapters and regulators of this system is important to substrate selection, my lab established a CLIP-C trap that was used to be able to identify potential regulators and adapters of this system. The CLIP-C1 trap is basically a stabilized version of CLIP system that I just showed you. Mutations in the chaperone CLIP-C prevents ATP hydrolysis. And so the adapters and substrates can bind to the chaperone, but the substrate is not able to be unfolded or degraded, which allows us to see adapters and regulators that interact with the system that would normally leave after the substrate is degraded. So based on affinity purification and mass spectrometry experiments previously conducted in my lab, UVR2 and UVR3 were identified as being potential interactors with the CLIP-C1 stabilized system, meaning that UVR2 and UVR3 are candidates for regulating CLIP proteolysis. These proteins are especially of interest as UVR2 and UVR3 contain a UVR motif, which are also found in 3 proteins that we've already identified in the chloroplast CLIP protease system. These proteins are the chaperones CLIP-C1 and CLIP-C2, as well as the adapter CLIP-F. As UVR2 and UVR3 contain a UVR motif, these proteins show promise in having a potential connection to the clip protease system. And in bacteria, UVR motifs mediate protein-to-protein interactions, which is why these proteins were chosen for study. The overall hypothesis for the study was that UVR2 and UVR3 engage the clip protease system as either regulators, adapters, or substrates. My specific aims were to try to determine if these proteins serve a redundant function and stress response, as well to test whether UVR2 and UVR3 form a dimer and interact with other clip proteins in vitro. So here we have an alignment of UVR2 and UVR3 amino acid sequences, showing that it has a very high percent similarity of 81%, suggesting that they have a potential redundant function. Here we have an mRNA co-expression network based on available microarray and RNA-seq data. So a connection between two genes in this network means that those genes were expressed at the same time under the same conditions. For UVR2 and UVR3, they are each other's top co-expressors, suggesting redundant function. Additionally, UVR2 and UVR3 co-express with ubiquination and autophagy pathways, suggesting a role in stress response, potentially like a role in retrograde signaling from the chloroplast to the nucleus in order that often occurs in stress conditions. To test my first aim that UVR2 and UVR3 have redundant functions involved in stress response, we created a double null line in order to carry out redundancy and stress tests. So single null tDNA lines of these genes were obtained from the Arabidopsis Biological Resource Center. tDNA is a huge DNA sequence that's inserted into the gene. And so you can see here where they were inserted for these genes. And that disrupts the expressions of these genes. These selected null lines were verified as null for these genes by using RT-PCR. To create a double null line of both UVR2 and UVR3, where they're both not expressed, we cross these genes. The pollen from one single null line, in our case was from UVR3, was used to pollinate the flower of the other single null line, in our case UVR2. Then the seeds are harvested from that and planted, creating the F1 generation. If the cross was successful, then the F1 generation will be heterozygous, which I verified using PCR. I then harvested and planted the seeds from that generation to create the F2 generation, in which 50% is heterozygous, 25% is wild type, and 25% is homozygous for both insertions. We selected specifically the ones that are homozygous for both insertions, as we would like to have a complete knockout of both genes' expressions. I used PCR to genotype each generation of the cross. Here is one of my gels that I used to determine which plants contain the UVR2 insertion for the F2 generation. Gene-specific and tDNA insertion-specific PCRs were completed for both UVR2 and UVR3. This is to make sure that we find those plants that have only the tDNA insertion bands for both of UVR2 and UVR3, but no longer contain any of the wild type bands remaining. This is to get a homozygous plant for these genes that means a complete knockout in expression. Through the genotyping I conducted, I identified a double null plants, which I harvested the seeds and will continue planting to establish a double null line that can be used for future experiments. This double null plant shows no phenotype, though, when in comparison to the wild type plant. However, the UVR2-1 single null line showed a potential germination phenotype during this process. On the plate that did not contain sucrose, 0% of the UVR2 null line seeds germinated. However, on the plate with sucrose, 22% of the UVR2 single null lines seeds did germinate, in comparison to 100% for the wild type and for the UVR3 single null lines that we were growing on the same plates. While there was no phenotype seen in the double null line, that means that we need to verify this phenotype by looking at future generations of the double null line that I established. As I mentioned earlier, UVR2 and UVR3 have no known function, but they do contain a predicted UVR motif. They also contain a DUF151 region, which also has no identified function. These proteins containing UVR motifs is interesting because UVR motifs mediate dimerization in bacteria, as well as in some bacteria, the activation of chaperones is regulated by UVR motifs. As I mentioned before, ClipC1 and ClipC2 chaperones and the adapter ClipF also contain a UVR motif, which is why these proteins are of interest to us, as we know that the UVR motif of ClipF can interact with the domain of ClipC that contains a UVR motif as well. To test my second aim, that UVR2 and UVR3 are heterodimers and interact with other CLIP proteins, we generated recombinant UVR2 and UVR3 proteins, which were then going to be able to be used for pull downs to check potential interactions with other CLIP proteins. To generate tagged UVR2 and UVR3 proteins, I used restriction cloning, in which restriction enzymes cut at designated spots, creating sticky ends around the gene sequence. There's also sticky ends in the plasmid that the gene sequence will be inserted into, meaning that the gene sequence will be inserted into the plasmid using ligation. I created 4 recombinant proteins: UVR2 with a his tag, UVR2 with a GST tag, UVR3 with a his tag, and UVR3 with a GST tag. And adding these tags to these proteins allows for us to complete affinity purifications and pull down assays in the future. Once the plasmids were created and protein expression induced in E. coli, I completed immunoblotting to check the expression of these proteins. I ran soluble protein extract from bacteria on a gel and then transferred it over to a membrane. Then I used an antibody that was specific to the tags on the proteins in order to visualize the proteins. So you can see from the results of the immunoblotting that for both UVR2 and UVR3, the recombination was successful, and I successfully created UVR2 and UVR3 recombinant proteins. Using the crystal structure of UVR2 of UVR motif interactions in bacteria, I made homology models of UVR2 and UVR3 potential interactions. I was able to recreate the interaction that is seen in bacterial UVR motifs, binding that usually bind and interact between the UVR motifs for bacteria. So it's possible that UVR2 and UVR3 could interact in that same way. So you can see here that I was able to recreate a heterodimer with UVR3 and UVR2, and as well as a homodimer with UVR2 with itself and UVR3 with itself. I was also able to recreate this interaction seen in bacterial UVR motifs with UVR2 and UVR3 with ClipC1. And this is why we think that UVR2 and UVR3 are promising candidate regulators of the chaperone ClipC1. So with the recombinant UVR2 and UVR3 proteins that I established and we grew in bacteria, we will be able to test this interaction in vitro in future experiments. So a quick summary of what I've done so far and what research still needs to be done. So I generated recombinant UVR2 and UVR3 proteins and checked the protein expression levels. I also genotyped the F2 generation and established a double null line. Then I modeled UVR motif interactions for UVR2 and UVR3. Still to come is to complete stress tests using the double null line that was established to see if there's a potential role that UVR2 and UVR3 plays in stress response, as well as to complete pull down interactions in vitros of UVR2 and UVR3 recombinant proteins to see potential CLIP protein interactors that UVR2 and UVR3 could interact with. I would like to thank my mentor, Marissa, as well as all the other members of my lab for all the help they have provided me this summer, as well as this program and the funding sources that provided me the opportunities all this summer. Thank you.